Good morning. I'm Dr. Akshay Panchal. I'm Assistant Professor in Pathology at Parul Institute of Medical Sciences and Research. Today I'm going to tell you something about staining in hematology. Okay. So you all know about peripheral blood film. Basically, a peripheral blood film is the smear which is made from the venous blood. Okay, that is peripheral blood. We make this smear on a clean glass slide. Okay, and then fix it and then stain it to see the various components of the blood under the microscope. Okay, various components of the blood under the microscope. The preparation of the smear is done, then there is a fixation of the smear. Fixation of smear is done using 100% methanol or 100% ethyl alcohol. Okay, acetone rich or water rich water mixed methanol or ethanol cannot be used. Okay, only 100% ethanol and methanol can be used. So this, after this fixation, the cells are in the we arrest the cell in their state, and we adhere them to the glass slide. Okay, basically, and their all the morphology is maintained due to this fixation. Later on, we stain it with using different Romanovsky stains. Okay, the indication for peripheral blood film in hematology, it is one of the basic, it is the most basic, it is most basic test in hematology that a hematology that a pathologist depends upon. Okay, it's a very basic test. Even before all the machines and all the advances in the medicine, okay, the peripheral blood film was the very basic source of information that could aid the clinical diagnosis. Okay, here for many diseases such as anemia, okay, whether it is nutritional anemia or whether it is hemolytic anemia or other sorts of anemia. Okay, some hemoglobinopathies can be drawn. We can draw some conclusion from the peripheral blood film. Some blood malignancies, hematological malignancies such as leukemias and lymphomas can be diagnosed and further investigated through peripheral blood film. Okay, certain parasites, for example, malaria parasite, filaria parasites. Okay, this can be easily visualized under the light microscope using peripheral blood film, which is properly stained. So this staining plays a very basic role and very important role in the process of diagnosis. Okay, so first you prepare a smear. You take a very thin, very tiny drop of blood on one end of the glass slide, and you spread it using another glass slide and forming a thin smear. Okay, so this is an example. This is an example of a very neatly prepared peripheral blood smear. So you kept a drop at this low end, and using another slide, just like this. So using another slide, okay, we just make we make a with a slow, with a fast and a thorough motion. We slide forward and make this smear. Okay, some precautions has to be taken, like not to apply too much pressure. Okay, the angle of the smear of the other slide that we are using to make the smear should be around 45 degrees. Okay, and not too we have to prepare a not too thin smear and not too thick smear. Okay, so if it is a thick smear, basically all the cells will overlap and we cannot comment upon the morphology properly. Okay, so and if it is very thin smear, then again it may give us some false data regarding the total counts. Okay, so this is very thin smear will happen if we are applying too much pressure. So what will happen, that there will be tailing basically. If you see if you see this smear, this is a kind of a very thin smear, and you may see that there are some sorts of serrations at the tail end of the smear. This is the tail end of the smear, this is the head. So what is our viewing area? Viewing area is just here. Okay, so it's near the tail end. Okay, just where all the RBCs are touching each other, and more than 50% of RBCs are not overlapping each other. Okay, here we can comment upon the morphology of the red cells. We can comment upon whether there is any parasite. We can search for any parasite, the morphological forms of the parasite. We can comment upon the whether, for example, whether it is a gamete of the Plasmodium vivax or a gamete of the Plasmodium falciparum, whether it is ring form or whether it is a tropozoid form. Okay, so all these kind of observations can be made. Here, if you are applying too much pressure, what will happen? That it will give us false differential count. Basically, what will happen? The larger cells that are neutrophils and monocytes, they will all accumulate at the tail end. So this is called tailing of the neutrophils and monocytes. So if we are viewing the smear here, we will not be able to find. We'll be seeing lesser number of neutrophils, which lesser than what actually are. Okay, so that is a thin smear. Now here is an example of a thick smear. So thick smear, what we are doing is we are taking a drop of blood, using the corner of one slide. We are slowly rotating it in circular motion, and to make a coin shaped area of approximately 1 to 1.5 centimeters. Okay, now let it air dry. Okay, and then we stain it. So this thick smear is used to it is a more sensitive process to sensitive test to find out the parasite. For example, malaria parasite. So if because here what is happening, lot of cells are overlapped. What we are doing here is we are preparing the smear, then we are uh, breaking out all the red cells. What we are doing is we are dipping it into water. So what is happening? All the cells are being, all the cell walls are being broken, all the hemoglobin is being washed out. Okay, only and only the the parasites and the nucleus parts of the cells will remain, and all the cytoplasm will get washed off. So we will be able to see the parasite very clearly. Okay, so this is the thick smear. Now basically what is happening, that after preparation of the smear, we are fixing it by dipping this smears into 100% methanol or ethyl alcohol. Okay, you keep it for 2 to 5 minutes for a proper fixation. If it is a humid environment, take care that we have to replace the methanol 2 to 3 times per day if it is a humid environment, because what will happen, that will be there will be some dilution which is happening slowly of the methanol, that is 100%. Okay, now types of these stains are there. Types of Romanovsky stains are there. These include field stain, Giemsa stain, Leishman stain, okay, Wright stain, etc. Of all of these stains are Romanovsky stains. They are basically metachromatic stains. Okay, metachromatic stains meaning they will impart different color to different components of the cell. Okay, and different types of cells. So that is the whole purpose here. Okay, we need to identify the red blood cells. We need to identify the nucleated cells, which are the WBC. We need to identify the granules in the cytoplasm. Okay, which will separate the WBC into granulocytes and the lymphocytes. Okay, so granulocytes include neutrophils, eosinophils, basophils. Okay, which contain this coarse, fine to coarse granules, which are azurophilic. Azurophilic meaning they are easily they have this affinity towards the acidic dyes. Okay, of all these stains, what we are using commonly at our laboratory is the field stain. Okay, field stain contains two components. One is eosin Y. Okay, that is the acidic dye. This acidic dye will stain the basic components in the cell. The basic components are these granules in the cytoplasm, the endoplasmic reticulum, okay, etc. And here is the field A, that is methylene blue. The component of this field A is methylene blue. So methylene blue is a basic substance. It will bind to the component of the cell. The acidic component cells are the nucleic acids, the DNA and the RNA. They basically form the nucleus. So this particular uh, stain will stain the nuclear component of the cells. Okay, so the procedure is very simple. What we do, see, basically the staining procedure for each and every stain has to be calibrated according to the individual laboratory environment. Okay, there is not one specific method, okay, or one specific as far as the timing of the staining, various steps of the stainings are concerned. Okay, those have to be calibrated according to individual laboratory environment. Okay, uh, but the basic concept, I'll tell you the basic concept. Basic concept is, first, in a smear that is fixed in methanol, after it is air dried, what we do is we dip the slides, fixed slides, into the eosin Y, that is the field B stain, for a time of 1 to 2 minutes. Okay, or if we are using a dipping method, so just like this dipping method, if you have filled this stain into a Coplin jar, what we do is we manually dip it in the eosin stain. So if we are dipping it, we from up to 10 to 15 times we dip into eosin Y stain, then we wash it off under running water, wash it off the stain completely under running water. After that, we dip it in the field A stain, that is methylene blue, for twice as much as time as we kept for the eosin Y stain, that is the field B stain. So this is for the time period for this is twice as field B stain. So if you have done 2 minutes for the eosin Y stain, then then after washing it in tap water, we will dip in the methylene blue stain for 4 minutes. Or if you have done 10 dips in eosin Y, then after washing off in the running tap water, we will do 20 dips in the field A stain, that is methylene blue. After that, again wash it in running tap water and let it dry. After drying, this slide, okay, these are the examples of very nicely smeared and nicely stained slides. Okay, so this should be the ideal shape of the slide, tongue shape, and in the distal 1/3, okay, distal 1/3 area, that is our viewing area. Like I said, here most of the RBCs are separated, and either they are just touching each other. Okay, they are not more than 50% of RBCs are not overlapping each other. Okay, so here in this viewing area, we are able to comment upon the morphology, different types of cells. We are able to find out the morphology of the parasites which are there. Okay, so